honor to be here to present our recent data about DADA2. Almost 10 years have passed since we started to study the immunophenotype of the first 2 patients of DADA2 diagnosed in our institute, and many basic studies have been carried out. It has become clear that DADA2 mutation impacts on both in function of innate and adaptive immunity, and these 2 arms interact very closely. Initially, in our laboratory, we were interested on the clinical aspect of immunodeficiency, so we set out to study the B-cell compartment, whereas more recently, we focused on the inflammatory aspects, and in particular in netosis and neutrophils, a new mechanism elegantly demonstrated in the publication of 2019 in Blood. NETs can activate autoinflammatory and autoimmune responses by different ways. They can break self-tolerance by being a source of autoantigens for autoantibodies, for example, in autoimmune diseases, in RA, in SLE, in ANCA-associated vasculitis. They can accelerate the inflammatory response by acting by a complement activation or the inflammasome activator. They are also DAMPs, danger-associated molecular pattern, and moreover, they can influence and activate other immune cells such as B cells, antigen-presenting cells, and T cells. Additionally, impaired mechanism of NET removal can accumulate active NETs and their component in the blood. So, NET may act as a regulator of inflammation and autoimmunity. They serve as biomarkers as well as the potential target for future therapeutics. Our idea was to investigate if NET disease in DADA2 are disease-specific and if they are influenced by the inflammatory microenvironment, and to discover new dysregulated pathways that explain how NETs can contribute to the disease. So we evaluated netosis in 22 patients followed by our institute, and in particular, we purified the neutrophils from peripheral blood. We stimulated them with adenosine, PMA, and TNF-alpha, and we analyzed netosis by imaging flow cytometry. Then we analyzed the mechanism of removal by ELISA assay in the plasma of patients, and characterized NET proteins by proteomic approach. Finally, we decided to investigate the crosstalk between neutrophils and dendritic cells, try to determine if NETs can activate inflammatory pathways in dendritic cells. Our cohort of patients consisted of 22 patients with genetically confirmed diagnosis, mostly of them with inflammatory and vascular involvement. Almost 80% were treated with anti-TNF, and they were all in clinical remission. We compared DADA2 patients with healthy controls and disease controls that are non-monogenic vasculitis, particularly patients with PEN. Disease controls included childhood polyarteritis nodosa and adults with idiopathic polyarteritis nodosa. They have normal DADA2 activity. They were tested while on treatment, and they are all in clinical remission as for DADA2 patients. Most patients with vascular manifestation fulfilled the criteria for including histopathological and angiographic feature. So we evaluated netosis by reproducing a published method that identify netotic cells by image stream flow cytometry. In fact, we analyzed the features of both suicidal netotic cells, which are spherical with the nucleus decondensed and with the colocalization with NPO and nucleus, and vital netotic cells characterized by elongated morphology with the condensed nucleus polarized within the cells. And we confirmed an exacerbated netosis in DADA2 neutrophils. We did not observe a strong effect of adenosine in vitro, but this could be due to a different method of analysis of netosis. Moreover, we observed a trend of increasing vital netosis. Then we evaluated the net removal mechanism that physiologically turn off the process, and we assessed by ELISA the amount of DNase 1 that is normal and DNase activity that is significantly reduced in DADA2 patients, but surprisingly also in PEN patients. The addition of a protein binding a possible inhibitor of DNase rescues the activity, rescue the activity in a significant manner, indicating a possible presence of inhibitors of DNase in the blood. Then we quantified ROS in peripheral blood, in neutrophils by flow cytometry, and they increased significantly in DADA2. So we decided to characterize NETs protein by proteomic approach and to discover new dysregulated pathways in DADA2 NETs. And despite the considerable overlapping between protein identity between 3 clinical groups and 4 stimuli, we multidimensional scaling analysis identified 2 distinct clusters, one consisting of DADA2 samples and the other one of all healthy donors and PEN samples. We identified 1,770 proteins. After normalization, differential expression in differential protein differences in protein expression were detected by ANOVA test. It is clear that DADA2 neutrophils show a peculiar NET signature in which a cluster of proteins significantly are upregulated and downregulated. Gene ontology identified different pathways that are enriched in DADA2 NETs. In particular, the most represented and highly significant is myeloid cell activation involved in immune response. These are the proteins including in this pathway 27 proteins, and among these proteins we found also MNDA, that is a transcription factor that control IRF7 expression, so linked to interferon pathway. Other pathways that are enriched in DADA2 NETs are actin polymerization and depolymerization, and regulation of actin polymerization and depolymerization, including a cluster of proteins downregulated significantly in DADA2 NET. This could be strictly correlated with the netosis process because regulation of actin regulation is important for co-rupture and extrusion of NETs. Moreover, the pathogenic mechanism of netosis has been demonstrated in disease in which mutated proteins are cytoskeleton proteins, for example, Wiskott-Aldrich or Papa syndrome. Moreover, we performed a co-expression network topology, an unbiased analysis that allowed us to identify biological processes significant in DADA2 NETs. When we compare DADA2 NETs with normal NETs or PEN NETs, we found a significant pathway regarding immune response, in particular type 1 and type 2 interferon and myeloid cell differentiation. On the other hand, when we compared normal NETs versus DADA2 NETs, we found a network significantly expressed that is linked to a regulation of TNF production. confirming that NETosis could be implicated in the dysregulation of TNF-alpha. When we looked at a cluster of proteins significantly modulated in PEN-NET with respect to the other groups, we found a cluster of protein down-modulated in which the pathways fibrinolysis, negative regulation of blood coagulation, and negative regulation of hemostasis are enriched. This could indicate that the cellular processes involved in the clot formation are more active, whereas the fibrinolytic phase is less active, resulting in a prothrombotic effect. Reactome analysis confirmed that fibrinolysis is a pathway enriched in PEN-NET with respect to DADA2 nets. These pathways are represented in all the stimulatory conditions in PEN neutrophils, indicating a possible signature disease-specific for PEN. If we consider the nets purified from adenosine-stimulated neutrophils, we found a cluster of upregulated protein in which the pathway endopeptidase activity is enriched. These proteins are co-expressed significantly, and the proteins involved in this pathway are this protein. Similarly, when we look at protein of nets isolated from TNF-alpha stimulated neutrophils, we found a cluster of proteins upregulated in which the enriched pathway, one of the enriched pathway, is cellular response to cytokine stimulus, and including a protein such as STAT1, IFI35, and IFI-T3. This thus suggesting that it could be a reciprocal activation between the TNF-alpha pathway and the interferon pathway. As we found as a significant pathway activated in DADA2 net myeloid cell activation, we analyzed the maturation of dendritic cells by flow cytometry by testing inflammatory and maturation marker on dendritic cells and cytokine production after stimulation in vitro with nets. As you can see, the dendritic cells from patients mature properly, but they are less active in producing pro-inflammatory cytokines. However, DADA2 nets seems to have a greater effect of stimulatory effect on the production of cytokines, on both dendritic cells from patients and normal dendritic cells. So I shall conclude by reporting how this work has been useful and what the future prospects are. NETosis, we reinforce the idea that NETosis is dysregulated in DADA2 patients and is implicated in the pathogenesis, but it may be also involved in the inflammatory process of polyarteritis nodosa. But with different effects downstream of NETs. Protein signature indicates that NETs are disease-specific, but the microenvironment may be a modifying factor. Moreover, NETs are a regulator of inflammation, not only activating macrophages, but also dendritic cells. And finally, differentially expressed proteins suggest that the interferon pathway may be linked to NETosis. Once validated and confirmed, these proteins significant in an independent cohort, these proteins could be a possible target for the future therapeutics. Thank you for your attention.